Our next speaker is Erika from the National Center for PTSD in Boston.
All right. Well, thank you so much for having me here. I'm going to be talking today, it's going to dovetail really well with what Caroline was just speaking about. I'm going to be talking about measuring trauma exposure and particularly how we measure traumatic stress and really making the case for trying to move beyond case status in our genetic association studies. So, okay, here we go. So there have been incredible technological and scientific advances in genomic medicine and unprecedented collaborative efforts to identify the pathophysiology and potential new biotherapeutics of psychiatric conditions. However, despite all this kind of remarkable sophistication and integration of knowledge across the omics, we are still too often left in the field of psychiatric genetics with just trying to predict simple diagnostic case status. I'm going to be speaking about PTSD today. That's my area of research, but really this concern applies much more broadly across psychiatric conditions. And it's not just the problem of overuse of case status that makes it difficult to accurately identify the genetics of psychiatric conditions, but it's also the case that psychiatric diagnoses are defined by an ever-changing list of symptoms that, while empirically informed, are ultimately decided by experts in a boardroom, sometimes resulting in hybrid models that might include some concessions to various investigators, but that are probably pretty far removed from the end product of the gene. Compounding this problem is the overuse of self-report measures with questionable validity and really substantial biases that will ultimately reduce our signal-to-noise ratios. So I'm going to be talking about these concerns today because ultimately our genetic association studies are really only as meaningful as our measurement of the phenotype is valid. And this becomes all the more true when we need to harmonize phenotypes across studies. So I really want to make the case for optimizing variables on both sides of this equation. Listed here are some problems that I'm going to touch on briefly today and try to give you a bird's eye view of the problem and what we might do differently to address it. So to start, let's return to that topic about the dichotomization of a continuous construct and why that's such a problem. I was going to show you some of this evidence from my own data directly, but in the coronavirus age, some of my own data sets are in lockdown and not accessible to me right now. So I'm going to focus on this classic reference by McCallum et al. On what happens if you dichotomize dimensional constructs. So for the case of PTSD, really there's no question in the literature that the construct is dimensional. So by this I mean individuals differ from each other by degree of symptom severity ranging from low, mild, moderate, and high symptom severity, but they don't differ from each other categorically or by kind. And what you can see from this McCallum text is what happens if you dichotomize a continuous construct. So no matter how you slice up the sample, whether you use a diagnostic criteria or some type of relative sample kind of midpoint, like a median split or something like that, is that you're actually going to always wind up taking samples from subjects who are very similar to each other, like subjects B and C in this example here, and separating them out into different groups artificially. And then you're going to take subjects who are actually fairly dissimilar to each other, so like subjects C and D in this example, and you're going to put them in a group together. So you really have substantial loss of information when you do this and reduce score reliability, because as you can imagine, if I were to take this sample 10 different times, subjects B and C might flip back and forth across the division line numerous times, and I'm just going to grab one snapshot of them and put them in one group when really their scores might sometimes get categorized in one group versus another. This also means you're constantly using an arbitrary or changing threshold over labs and measures and over time. And it ultimately results in reduced effect size because even if you have sufficient sample size to run your analysis, if you dichotomize a dimensional construct, simulation studies will demonstrate that you actually have a reduced effect size in your results. And on the flip side, if you actually run your analysis in a very small sample, the simulations will show you that you will actually have an increased effect size, but that's an artificial, so it's an invalid result when you're running small samples and you're dichotomizing a dimensional construct. All of this is going to result in poor construct validity and less likely to replicate results across studies and result in reduced statistical power. And in fact, again, simulation studies demonstrate that if you dichotomize a dimensional construct, it's actually akin to reducing your sample size. So nobody wants to do that. And of course, all these concerns become magnified when you need to correct for multiple testing. So what can you do? Well, the simple thing to do is to ditch case status altogether, or at the very least, supplement it with the analysis of continuous severity scores, or even symptom counts of the number of symptoms that are present, if necessary. And this is really simple, but it yields improved validity and statistical power for your analysis. However, we can actually do better than this, because even with dimensional scores, there's still the problem that the disorders that we're studying are defined in a top-down approach rather than a bottom-up or even a genetics first approach. So this is that boardroom problem that I mentioned earlier. So one way around this is to refine our phenotypes by saying essentially, okay, all these diagnoses are imperfect measures of psychopathology as it exists in the real world or in nature. And when we combine these imperfect measures to try to get at what's common across them, we may actually have a better reflection of what's out there in nature, perhaps more of an endophenotype that may be closer to the end product of the gene. So one way you can do this is with latent variable analyses, particularly exploratory and confirmatory factor analyses, which are developed for modeling dimensions. And these latent variables are indicated by what's common across imprecise indicators, and as such are free from measurement error and have improved construct validity and statistical power. So I thought I'd give you a few real world examples of this from our lab. So first, if you just want to focus on a disorder like PTSD, you might wonder about whether no PTSD is really the end of the dimension. That is to say, maybe the dimension doesn't range from no PTSD to high PTSD symptom severity, but rather from adaptive functioning, or what you might call resilience in response to a traumatic experience, to high symptom severity. So in our lab, we conducted a twin study to look at this issue, and we showed that when we use factor analysis to combine PTSD symptoms with a measure of resilience, we actually did a better job of representing the whole spectrum of responses to trauma exposure, again, incorporating both adaptive and non-adaptive responses. And we increased the heritability coefficient for this broader spectrum or latent variable of responses to traumatic stress, compared to the heritability coefficients of either PTSD severity alone or resilience alone. So by simply focusing on PTSD in our molecular genetic association studies, we might actually be limiting our ability to identify genetic markers because we simply don't have the full range of the construct adequately represented. And we have reduced score variance and therefore reduced genetic liability for these variables. We can also move beyond individual diagnoses to look at how psychiatric diagnoses map onto broader spectrums, such as internalizing and externalizing dimensions, which again may be closer to the end product of a gene. In our lab, we've shown that PTSD is associated with genetic risk for both internalizing and externalizing, and therefore it's possible to study the genomics of broader common factors like internalizing and externalizing, or the sort of personality traits that might underlie them, like negative affectivity and impulsivity, respectively. And again, this would improve the representation of the phenotype, as it's unlikely that the genetics of any single given disorder are all that distinct from the genetics of a highly comorbid psychiatric disorder. And we have plenty of evidence of this from the results of cross-disorder genomic studies and bivariate score regression. So we can model what's common across psychiatric disorders and hope to thereby have a better signal. But after we account for those common factors and the genetics of those common factors, we can also use a latent variable analysis to account for what's left over. So this would be asking, well, what are the genetic factors that make disorders or individuals with the same disorder unique from each other, distinct from each other? So we can get more into the weeds and examine, for example, individual symptom factors like the PTSD symptom clusters after accounting for, say, the overlap between PTSD and depression and anxiety and other internalizing disorders. Or we can have a unique, look at individuals with a unique clinical presentation, such as those with the dissociative subtype of PTSD. So our lab has shown that some individuals have a really unique presentation of PTSD defined by comorbid dissociative symptoms. And if we don't kind of adequately account for that level of heterogeneity in the clinical presentation of our subjects, well, then we may be actually hindering our search for a signal. There are several other advantages to use for latent variable analyses. I won't get into them, but it's a very flexible approach and has a lot of sort of already established protocol to help us get through determining what's the right measure for, or what's the right model for the data. And if you model things with your latent variable analyses, you typically will improve your statistical power and the magnitude of association. However, there are some caveats that should be noted. First, one thing is that classes can be deceiving. You might think, well, if I can't just dichotomize my continuous construct, perhaps I can use a latent class analysis to really get at what is the true class or the true group of people with the diagnosis and study the genetics of that. But when you do that, what happens is if you run a latent class analysis on a dimensional variable like PTSD, you get groups that differ from each other or classes that differ from each other by low, moderate, and high PTSD severity. And that's what we refer to as a salsa pattern, because people are mild, medium, and hot on your construct. And it's actually an indication that you've got a dimension in your phenotype rather than classes. So if you try to run a latent class analysis on that high PTSD severity group, you've essentially kind of got garbage in your phenotype, and you're not going to really have adequate or valid genetic association results. Another thing I've heard or seen people talking about is maybe using network models as potentially the way to go to identify the most important central symptoms from a diagnosis and then study the genomics of this. But I would strongly caution against this, as network models, particularly of cross-sectional symptoms, are highly unreliable and more likely than not to reflect a lot of measurement error. So what can we do to address this boardroom problem? Well, first is to not be restricted by our DSM-defined disorders that are likely far removed from the end product of the genes, and then to try to use factor analyses to refine our phenotypes and measure common features of psychopathology to obtain a broader coverage of the spectrum. And then after accounting for the genetics of those common factors, we can look at what's left over using a model that best represents the data. So I want to now turn briefly to talk about the quality of the measurement. And self-report measures that we use a lot of the time in our genetic association studies are low cost, easy to administer to lots of people, but the problems that have been associated with them have often been discounted. People seem to think that they're good enough, but in fact, self-report measures are subject to substantial biases, such as yay-saying or over-reporting, and sometimes it might not be clear whether you're studying the genetics of, let's say, PTSD or the genetics of an over-reporting response style. So this becomes problematic. There's also plenty of evidence that individuals without criterion age traumatic events can score above the cut point on our measures of PTSD. So it's not really clear what we're measuring at that point. Our traditional self-report measures of PTSD don't include validity scales to address these kinds of concerns, include pretty nuanced symptoms that you're asking people to differentiate between, say, re-experiencing a traumatic event versus anticipatory anxiety about the consequences of a traumatic event. And if you look at the diagnostic accuracy of our main measures of PTSD, self-report measures of PTSD, they're really not that great in comparison to clinical interviews. So as a result, we also haven't used most of our PTSD measures, I should mention, haven't been developed using really classical test construction techniques, which are well-developed literature for how to develop measures that would have better psychometric performance. So these are really concerns that I have about the use of self-report measures with PTSD. So what can be done beyond just choosing the most well-validated assessments? I think it is important to budget time and money for a more comprehensive clinician-administered assessment of PTSD, at least for a portion of the sample, so that you can see if your genetic association studies line up across your self-report and clinician-administered measures. So finally, I want to talk about one more concern, which is third variables as serving a role that's more than just as the covariate. So third variable problems or confounds are often included in our models of PTSD or the genetics of PTSD as just a sensitivity kind of analysis to see if our results still hold. However, that might not be inadequate, because it's important to note that actually in many cases, variables like trauma exposure and demographic variables, traumatic brain injury, other comorbidities are actually moderating our effects of interest in a G by E kind of way. And so we actually may be throwing out good effects if we, I'm getting messages here, sorry. If we may be throwing out good data, good genomic effects if we don't look at these interaction effects. So one of the concerns here is that interaction terms are typically quite small in terms of effect size and require really sufficient sample sizes and adequate power in order to examine them. But if we don't look at these interaction terms, we actually may miss important subgroup effects. So for example, if you don't look at, let's say, trauma load by genotype and predicting PTSD, well, you might miss the fact that there could be genetics of PTSD for people who really have small trauma load. And so they maybe have more of a genetic predisposition towards PTSD when exposed to even low levels of trauma. And if we don't model that interaction term, we may actually miss that effect. So the moral of that story is really just to sufficiently power our samples in order to look not just at covariates and confounds, but also the interaction effects. So to sum up, the work that's being conducted now to study the genetics of psychiatric disorders is really moving at an incredible speed. And I think we can further advance the science of the genetics of psychopathology and develop new treatments if we invest the time and money and energy necessary to measure our phenotypes with the same level of quality control and precision that we demand of our genetic processing pipelines. So ultimately, doing things like working with severity scores, modeling broader phenotypes, investing in high quality assessments, and modeling potential interactions with third variables is high cost, but actually has high potential and clear potential for high reward. And I'll leave it at that.
Thank you so much. There are two questions. Andreas, do you want to go do this? Shall I? Okay, so there's one from Jack Hattemer for you, Erika. In using symptom dimensions for GWAS, how do you deal with the short time scales typically covered in surveys compared to lifetime diagnoses?
Yeah, that's true. Although there are, if you're going to look at symptom dimensions, we do routinely in our lab assess for PTSD symptom severity over someone's worst period of time. And so we're still getting a lifetime measure of symptom severity. So for example, we'll ask them to identify when their symptoms were at their worst and then anchor all of the symptoms to that one same time period.
Okay,
thank you. Go ahead. No, I was muted forcefully by the host.
Okay, sorry. And there's a second one. Has similar work been done with the tripartite assessment of PTSD?
Say that again, let me find the question.
It's the second one.
For the tripartite assessment in PTSD. Let's see, I'm not sure if you mean the tripart assessment as in there's personality measures that differentiate things like depression and anxiety, like splits between high and negative emotionality and positive emotionality, or if you mean things like internalizing and externalizing subtypes of PTSD. Oh, what's their question here saying? Psychophys?
Yes.
Yeah, well, no, we've done work looking at internalizing and externalizing subtypes of PTSD and the molecular genetic signatures of that. And we have actually found differences. So for example, in one of our early GWAS studies of PTSD, we found effects from the RORA gene on PTSD. And then when we looked further at that, we found that that was actually specific to PTSD with comorbid internalizing disorders. And it wasn't defined by, say, people, which are the people who usually are having high physiological reactions, and it wasn't defined by people who are having under reactions, less than expected physiological reactions, so trauma reminders, which you would expect in externalizers.
Do we have time for one more question? Because I would have one more.
Yeah, sure.
So for the class identified by latent class analysis, do we know if there is a difference in responsiveness to treatment, PE, psychotherapy, or pharmacological treatment?
Yes, so you're talking about the dissociative subtype that I mentioned? Yes, actually, so there have been a number of studies looking at that. It's been challenging because some of them haven't really been powered to look at whether the subtype would moderate treatment response. But one of our studies actually did show that individuals with the subtype showed a less steep slope in their response to standard PTSD treatments, psychotherapies. And then there's a more recent paper that showed that individuals with the subtype were less likely to respond to sertraline treatment than individuals without the subtype.
Okay, and there's two more questions. One is related. So advantages, disadvantage of latent variables over non-parametric machine learning. Kind of a guidance, what's the best method for the job? I guess that's kind of captured into questions here.
Yeah, well, I think some of it's going to have to do, when you start to talk machine learning, you really have to have the right samples and the right replication samples. And that's not always something that you can make happen in these types of data sets. The advantage of the latent variable model, I think, is that you actually have the fit statistics that will tell you whether you've adequately represented the data that's in your study, and it will even tell you, overall which model fits the best compared to other models, and it will tell you where there are parts of model strain where maybe you're underrepresenting or overrepresenting some associations in very specific parts of your model, and you're not going to get that in most machine learning types of analyses.